All right, sports fans, pre-AP biology. Today, we are going to cover probably one of the more complex things that um, I talk about all year. And that's a topic titled protein synthesis. Um, I'm going to provide for you all my lecture notes and obviously a copy of this video. I might suggest to you watching this more than once, or maybe watching the first, chunk of it, maybe 10, 15 minutes, and watching the rest of it. Because there's going to be a lot to absorb with this topic. Okay. So put your thinking caps on, folks, because this is going to be a rough ride because there's a lot to cover. I would suggest getting in a quiet area where there's no distractions. Take your phone and throw it in another room. All right, don't have it by you. It's only a distraction. Don't throw it. Just move it to another room. All right, let's review a little bit. The cell factory. This takes us all the way back to the first semester, right? We said that all cells build proteins. That's what they do. For example, red blood cells make proteins, muscle cells make proteins, sperm cells, bone, whatever. All these different cells make proteins, right? Hemoglobin is a good example of a protein that gets made by red blood cells. So some cells build the same proteins. Like all of these red blood cells here, they're all making hemoglobin. They all make the similar protein. Some cells make different proteins. Different cells make different proteins. For example, your liver cells, which aren't shown here, but liver cells make a protein known as catalase. It's a completely different protein, does something completely different from hemoglobin. If you want to know what catalase does, stop the video now and go Google search it. You guys are pretty good at that. So anyway, this is, I think, a pretty easy concept, right? Some cells, all cells build proteins, but they don't all build the same proteins. All right, this is important. What's needed to build a protein? If you're, it's like, if we're going to make chocolate chip cookies, right? We need sugar and milk and butter and all that stuff, right? But if we're going to build a protein, one of the things that we have to have is energy. Right? We have to have, your cells have to have energy or else they're not going to function, right? We talked about cellular energy, in the first semester. We called it ATP, adenosine triphosphate. This is cellular gasoline. Without it, your cells aren't going to operate correctly, right? They need to have energy. So, it's important that we eat food so that we can obtain that energy so that our cells can do what they do, right? They build proteins. Another thing your cells need is ribosomes, right? Ribosomes are the organelle that's responsible for building the protein. So if you don't have those organelles, it's going to be kind of hard to build those proteins. Amino acids, right? Amino acids are what the proteins are built out of, are built from. Does anybody remember how many different types of amino acids there are? There are 20 different types of amino acids, and we'll look at those in a little while. They actually all have different names, but these are the materials that are needed to build the protein. Of course, this is a big one, right? Your cells have to have instructions. They need the DNA. If you don't have the instructions, if you don't have the recipe to make chocolate chip cookies, it might be difficult to make them. But if you have the instructions, it's actually pretty easy, right? So DNA is the instructions. Our cells have to have that. Now, here's another one that we haven't really talked about yet. We're going to get into this in a couple of minutes. It's called RNA. Now, RNA is a nucleic acid. That's what NA stands for. So it's kind of similar to DNA a little bit. There's a couple different types of RNA that I'm going to introduce you to, but I'll explain what their functions are a little bit shortly. But again, in order to build a protein, it's a process, right? We talked about what do you start with, what do you end with, and how do you get there, right? There's some steps involved. So I want us to think before we get into this process, this is pretty much the starting point. All of these things are needed to build a protein. We're eventually going to get proteins that get built. That's our final product. So we have to, again, figure out how is the process, or how does it unfold? How does it happen? How does it work? All right, so let's take a look here at this picture. This is a typical cell. You've got your nucleus here. You got some organelles floating around out there. But I want you to notice these little dots. Okay, these little dots are ribosomes, right? And they're kind of scattered throughout the entire cell. There's thousands and thousands of these ribosomes inside of your cells. And we know what their job is, right? Their jobs are to build proteins. But ribosomes don't do their job very well unless they have the instructions. So they need these instructions in order to build their protein. So I want you to ask yourself right now, in this picture here, maybe even put your finger on it. Where are all the instructions in the cell? Well, if you're pointing to the nucleus here, if you're pointing to all these chromosomes, you're right. That's where all the DNA is, right? That's where all the instructions are to build the proteins. In fact, all the instructions to build every protein in your entire body are in there. Think of the nucleus and think of all of this DNA. Think of it as like a giant library. There's just a ton of information in here that your cells can use to build a whole bunch of proteins. Now, for example, if this is a red blood cell, is your red blood cells, do they need all the instructions in here to build every protein in your body? No, they don't need all the instructions. They just need certain instructions. It's like if you were going to go to the library and I don't know, let's say you had to write a report about the American flag, the history of the American flag. So you went to the Crown Point Library and you wanted to find a book or something about, the history of the American flag. I know nobody goes to the library anymore to check out books, but, back in my day, that's what you kind of did. So whatever, so you go to the library and you find this book, that's titled Everything You Need to Know About the History of the American Flag. There's a million books in the library, but you don't need every single one. You just need like that one book, right? So, that one book would come in really handy to write your report. Well, inside of this nucleus are all the DNA. Everything's in there, that your cells need. But again, your cells don't need everything. They just need certain pieces, right? They need certain books in order to write their reports or to make their proteins. All right. Now, there's a couple of issues that your cells have, okay? One of the issues is the DNA that's inside the nucleus, it can't leave the nucleus. It can't leave there. It's like, if you went to the library, you found your book, titled The History of the American Flag, and you go to the librarian and you're like, oh, I need to check this book out. And the librarian says, no, you can't, this book can't leave the library. It's a reference book or something. It can't leave here. It must stay in the library. But you're like, well, I got to have this book. I need it, to write my report. Librarian says, no, I'm sorry, you can't take it out. So that's an issue. The DNA cannot leave the nucleus, but your ribosomes have to have that information. They have to have the DNA. The ribosomes can't go into the nucleus either. So you've got this issue of, the ribosomes need the DNA, they can't go in, and the DNA can't come out. So we've got this real big problem, if your ribosomes are going to build protein. So how are the ribosomes going to get the instructions? Well, think about this book that you found, I mean, I don't know, you got a phone, maybe you can like take pictures of the pages that you might need, or maybe there's a copy machine in the library. You can make copies of certain pages of the book. Whatever. I guess you could steal the book, but that would be totally against the law. Don't do that. All right. But maybe copying some pages might be something a little bit easier to do that wouldn't get you in trouble. So of course we're going to go ahead and copy those instructions. So instead of the DNA leaving the nucleus, something can copy those instructions and carry them out to the ribosome. Well, that something is RNA. More specifically, It's called mRNA. It's a specific type of mRNA. But here's what it does. It says mRNA is responsible for copying the DNA and carrying it to the ribosomes. This process is called transcription. This word transcription, okay, it means to copy. The word transcribe means to copy something. So if mRNA can copy the instructions, that would be great. Okay. Now, before we get into some more of the functions of RNA, I want us to kind of take a look at the structure of RNA. Okay. In fact, either mRNA or tRNA, they're both kind of put together the same way. If you look at this picture here, okay, on the right-hand side, okay, this is just your normal DNA, right? This is your deoxyribonucleic acid. We've got our double helix, we've got our nitrogen base pairs, and we've got our sugar phosphate backbones, all that stuff. And we've got our cytosine, guanine, adenine, and thymine. This is the structure of DNA. We know this. We've memorized this by now. But over here on the left, this is RNA. And I want you to think, just by looking at the picture, what are some of the differences here between RNA and DNA? Well, one of the big differences that you see is that there's only one strand. There's only one strand of nucleotides, whereas DNA has two. So this is a single-stranded molecule. This is a double-stranded molecule. And another big difference is if you look at the bases. Right, notice how RNA has cytosine and guanine and adenine. But there's another base here that is not found in DNA. It's only found in RNA. It's called uracil. Uracil actually takes the place of thymine in RNA. Now, your base pairing rules would still be the same. I mean, C would bond with G if they could bond, and A would bond with U if they could bond, but there's no bonding here because this is all a single-stranded molecule. So Chargaff's rules would still be the same, except there would be no thymine. Okay, so one thing we have to know is we have to know some of the differences between RNA and DNA. So RNA is single-stranded, DNA is double-stranded. RNA, here's another big difference. DNA has that sugar, deoxyribose. RNA has a sugar called ribose. So this is called ribonucleic acid, not deoxyribonucleic acid. RNA can move in and out of the nucleus where DNA cannot do that. DNA can't leave. RNA has the four bases, guanine, cytosine, adenine, and uracil, where DNA has guanine, cytosine, adenine, and thymine. So you guys do need to know these differences, all right, between RNA and DNA. All right, now what I want us to do right now is to pause the video, maybe take a little break, absorb some of this information, maybe even watch this again before we go into the next part, all right, because the next part is pretty big. All right, so take a little break if you want to, go get something to eat, get something to drink, come back, and then we'll watch the second half. All right, welcome back, everybody, if you took your little pause. I've been using this diagram for about 25 years. So it's an oldie but a goodie, all right? And this is, you can kind of see the extent of my artwork. I'm not really that good at drawing, but I think it's simplistic enough to where we get the idea of what this is. So here's you, okay, up here. Yes, you are stick person, okay? Here are some of your cells. Yes, they're very simplistic. There's only 3 cells, but there's whatever, billions and billions of them in your body. But what we're seeing here is one of your cells, okay, just one of them. I didn't draw every single component of the cell. I didn't draw the endoplasmic reticulum and the Golgi bodies and all that type of stuff. But I do have a few things inside of this cell that should be in everybody's cells. Okay, for example, right, you see the dotted line here, right? This is our nuclear membrane. This is the nucleus of the cell, okay, right in here. And inside that nucleus, just like there should be some chromosomes in there. Now we know that there should be 46. I didn't draw all 46 in there. I just drew a couple of them, but there are 46 chromosomes in that nucleus. So what I've done here is I've just kind of circled a little tiny chunk there. That's DNA, right? All these chromosomes are made of your DNA. All right, so that's kind of simplistic, but whatever. These little squares here, these guys are your ribosomes. All right, they're really little. There's like millions of them in your cells, but whatever. Ribosomes, this is where your proteins get built, right? Those are the organelles that build the proteins. So protein synthesis, right? That's where it happens. That's where all the magic occurs, is on those ribosomes. Here's your mRNA, right? Here's our little single-stranded molecule, right? We just talked about this in the previous part of the lecture. These colored molecules here, these are amino acids. Now, amino acids, the reason why there's different colors is because there's different types of amino acids. Right? In fact, there's 20 different types of amino acids. These are what proteins are made of. So think of like a brick house is made of bricks. So we've got 20 different types of bricks to choose from. I probably didn't have 20 different colors, but I tried to use as many different colors as I could. All right, but that's what those are. And then finally, we have a structure called tRNA. It's kind of this triangular structure here. I'll explain to you what tRNA is as we go through this lecture, but kind of get familiar with all these different components. All right, let's move on here. Now, when it comes to building a protein, there are two different, processes. The first process, which I've already referred to, is a process called transcription. Remember what I said the word transcribe means? It means to copy. All right, and I mentioned this earlier that, the DNA, the instructions inside of the cell, they can't leave the cell, they can't leave the nucleus. So they have to get copied, right? So for example, in this picture, right, here's our little piece of DNA here. Let's just say that little chunk there, which has been kind of magnified up here, this is our piece of DNA, okay? That's what this little piece is right here. Let's just say that particular piece of DNA carries a very specific set of instructions for how to build a certain protein, okay? But again, those instructions there, they cannot leave the nucleus. They can't leave. So something has to come in and copy those instructions. Well, that's what our mRNA is for, right? This little piece of mRNA here, for example, that thing can go into the nucleus and copy those instructions. So that's what this is here. This is our piece of mRNA, right? Our single-stranded molecule, and it's going to copy the DNA. Now, this is where it gets a little tricky, so kind of stay with me here. All right, going back to our DNA here, all right? Notice how I've kind of put these bases in sequences of 3, right? We've got ATG, TTT, GCC, right? They're in these sequences of 3, and then on the other side of the DNA, right, is the opposite side, right? So opposite of A is T, opposite of T is A. So we have the complementary strand on the other side. So this is no different from what we've already learned. Now we do have a name for a sequence of 3 bases on DNA. They're called DNA triplets. So for example, ATG, that's a triplet. TTT, that's a triplet. GCC, on and on and on. These are like little code words, okay? They're like little pieces to the recipe. They don't make any sense to us, but they will to the ribosome when the ribosome builds the protein. Now, again, you'll notice that we have 2 sides of triplets. We have 2 sides of instructions. Well, the mRNA, since it's only a single strand, it can only copy one side or the other. It can't copy both sides. All right, so we have 2 terms here. One's called the sense strand, and the other is called the anti-sense strand. The sense strand is the side of the DNA that's going to get copied. Okay. The antisense is the side that doesn't get copied. And just, you know, there's no rhyme or reason to this, but I just chose this bottom row here to be our sense strand. Okay. So these triplets here are going to be the ones that the mRNA copies. Okay. Now, when the mRNA copies these triplets, it actually copies the opposite. So if TAC is the triplet, what would be the opposite of TAC? I'll give you a second. Well, the opposite of TAC, right? Opposite of T would be A. The opposite of A, now you might think it would be T, but you have to remember this is RNA. So the opposite of TAC is AUG. This is the copied version of the DNA. All right, this is what we call an mRNA codon. This is a codon. This is a triplet. Codons are on mRNA, triplets are on DNA. Then we have the next triplet, AAA. What's going to be the opposite of AAA? UUU. Okay, remember this is RNA. Then we have CGG. The opposite would be GCC. Then we have ATT. The opposite would be UAA. So this completes the process of transcription. Transcription is just mRNA copying the DNA. That's all it is. So if you understand this slide here, and understand what I've just been talking about for the last, 5-10 minutes, then you completely understand the process of transcription. All right, now I want you to pause the video here. All right, take a break, relax, go get a sandwich, get something to drink, check your texts on your phone, whatever. Take a break. Maybe even watch this part of the video again before we go into the next part. All right, welcome back from your break. I want to quickly review this. Okay. So again, what we've done during transcription, and this all took place in the nucleus, is the mRNA copied the DNA. That's all it is. Now we have to think, where do we go from here? Right? I mean, what's the purpose of copying that DNA? What is it in your cells that needs those instructions? Well, it's the ribosomes, right? Your ribosomes have to have the instructions so they can build the protein. So this mRNA, once it copies the instructions, then it's going to go to the ribosomes. So let's quickly review transcription here. Transcription takes place in the nucleus. Transcribe means to copy. This explains what happens during the process, right? DNA is copied by the mRNA. Only one side of the DNA can get copied. That's known as the sense strand. mRNA copies the opposite sequence as the DNA, right? It doesn't copy it exactly, it copies the opposite. A DNA triplet is 3 nitrogen bases on DNA. An mRNA codon is 3 nitrogen bases on mRNA. So there's some vocabulary there we have to know. All right, so now we're ready to go to the ribosome. Okay. Now, I want you to take a look here. There's a lot going on, but let's first take a look at this strand, right? This is the mRNA that's the same thing as this, okay? So that mRNA has now moved to the ribosome. So now we're at the ribosome. And the ribosome has to read these instructions, but there's a problem. The ribosome doesn't quite understand these words, okay? It doesn't really understand what they mean. So in order for the ribosome, something has to translate those words for it. That something is what we call tRNA. Now, the T, you might think it stands for translating DNA. It actually stands for transfer DNA. But I always tell kids, if it helps you remember what it does, then think of the T as translating, because that's kind of what it's doing. It's kind of coming up with a word that the ribosome will understand. That makes sense. Imagine like this is like written in Spanish, and you don't understand Spanish, right? You need something to translate it for you. Well, that translator would be the tRNA. Okay. So what the tRNA does is it takes the opposite of the codon. So what's the opposite of A? The opposite is U. This is still RNA. The opposite of U is A. Remember, A bonds with T, but U takes the place of T. The opposite of C is G. So this is what we call an anti-codon. This word here, the ribosome understands. Now, what does it mean to us? I'm going to show you how to figure out what it means. This is what we call a codon chart. I'm going to give this to you to use on quizzes and tests, but you need to know how to use it, okay? Now, before we use this thing, I want you to look at all these words on the outside, okay? All these words, glycine and aspartic acid and alanine and serine, whatever. These are amino acids, and there's 20 of them. If you count them up, you'll find 20 amino acids. Some of them are on here twice. If I can find some of those, but that doesn't matter. Okay, there's 20 total amino acids. So this is what your ribosomes need to know. They need to know how they're going to build the protein, which building blocks they're going to need. Now, since this is called a codon chart, we are going to use codons to help us figure it out. So our first codon is AUG. So let's take a look at this chart. Now, we're going to start on this inner circle. The inner circle, what's the first letter we have to use? A, right? It was AUG. So we're going to use this right here. And we're going to start here. And we're just going to move outwards. So here's A. Then we're going to go to U, right? So notice here's A. We can go to G, we can go to A, we can go to C, or we can go to U. We want to go to U. So A, U, and then what was it? G, right? So here's U. We can go to any of these four spots. So we're going to go to G. So A, U, G, and this is the name of the amino acid. It's called methionine. So that code word right there is a code for methionine. It's a specific ingredient that's needed to build this protein. So now the the ribosome knows what to get, and it grabs onto methionine. But there's methionine in your cells. Now this whole thing is going to shift over. It's going to move over to here. Here's our next codon, U, U, U. Now, again, your ribosomes don't understand that. So what would be the opposite of UUU? The opposite would be AAA. And that would be the anti-codon. But UUU, that's what we're going to use here, right? So here's a U, right? Start on the inside, and then here's our second U, and then here's our third U. So this is phenylalanine. We're just going U, U, U. So phenylalanine, here's our next amino acid, right? Then we move down to the next one, GCC. The opposite would be CGG, but we're going to use this codon here on our chart. So here's G, right? GCC. That's alanine. So there's the next amino acid. See how we're just kind of linking these together, okay? Then we get to the last one, UAA. UAA is a codon, right? The opposite of UAA would be AUU, but we're going to use UAA. So here's U, A, and then A. You'll notice that it says stop. Now stop is not a amino acid. It's simply a signal that says stop. Okay. Once the ribosome gets to that codon, then it knows to stop building this particular protein. All right. So what we just did was we made or we synthesized a protein. A protein is basically just a chain of amino acids. And these amino acids are linked together by what we call peptide bonds. Don't worry so much about the chemistry of that. It's another type of bond you have to know. But that's what links them all. Think about just like a links on a chain. Okay. Now, most proteins are like thousands of amino acids long. This one just happens to be 3. But again, an amino acid chain, it's also known as a polypeptide, many peptide bonds. It's also known as a protein. These 3 words are interchangeable. All right. Now, this whole process is called translation. You think about what happens, right? We've got our mRNA instructions, and they have to be translated so that the ribosome knows how to build this protein. All right, so a few things that I want you to know about translation. Number 1, translation takes place at the ribosome, and this is where the proteins get built. The mRNA codons need to get translated into amino acids. The tRNA is what does the translating, using those anti-codons. The anti-codon is the opposite of the codon. We can use the mRNA codons on our codon chart to look up the amino acids. You're going to have to be able to do that. Amino acids are linked together by peptide bonds, and a chain of amino acids is also known as a polypeptide or a protein. Okay. Again, I would probably watch that part of the video again to make sure it makes sense to you. Okay, now we're going to do a little bit of practicing. Okay. So we're going to fill this chart in, okay, just some practice. So let's say, for example, I give you a DNA triplet, okay, CCT. Let me get back out of this real quick. Oops. All right, so CCT. And I want you to tell me what the codon is, the mRNA codon. Well, just think of the opposite, right? What's the opposite of C? G. What's the opposite of C again? G. What's the opposite of T? A. So your mRNA codon would be GGA. Now, the anti-codon. Remember, this is tRNA. So the anti-codon is the opposite of the codon. So if you have GGA, we're going to have CCU. Remember, there is no thymine in RNA. Now, the amino acid. That's where we're going to use our codon chart. So as long as you know the codon, GGA, we can go to our codon chart, right? G, G, and then A, that's glycine. So our amino acid is glycine, okay? Now let's look at this next one. Now here I give you a little bit of a curveball. I give you a codon, CGA. We've got to come up with the other 3, okay? So let's do the DNA first, right? If it's CGA, what's going to be the opposite? GCT, right? Because there is thymine in DNA. The anti-codon, again, we're basing it off of this codon here. So GCU, right, would be the anti-codon. Now the amino acid. Again, we're going to use the codon to find the amino acid. So CGA, so here's C, G, and then A, this is arginine, okay, arginine. So you're going to have to be able to do something like this. go ahead and if I give you a chart, fill in the rest of the chart. All right, but there's a lot here, guys. I understand it. This is a big concept. So watch this again, watch parts of it again. If you have questions, you can email me or you can ask me during class. All right, guys, have a good day. I will talk to you